Hello students, welcome to lecture 5 of PCS Booster Course. So we are trying to highlight some important topics from prelims perspective for upcoming Punjab Civil Service examination. So topic for today's discussion is from General Science section. We'll be decoding a theme which appears quite occasionally in state services exam, and PCS is also not an exception. Already couple of questions have been asked in last 5 PCS examination based on the blood group themes. Now, this topic is not only important from conventional, you could say human physiology part, but also equally important from current affair perspective, because recently a new blood group was discovered in India and which was making headlines. So basically, once again, the question might be framed based on that new current affair theme, or typically when some particular topic is value added in such subjects, the examiner tend to ask the conventional question. So we will be highlighting all important facts related to blood groups, what are their different types, how are blood groups named, what are the ideally recipients and donor angle, and then the current affair part in the final slide. So without wasting any time, let's start with today's topic on blood groups. As you could see on your screen, blood group is not only limited to ABO, which we conventionally used in our day-to-day conversations, or you could say even though ABO continues to be universal blood group, but it's not the only blood group which is present. MN blood group, Bombay blood group, as you could see on your screen, Knobs, Duffy, Lutheran, Gerbich, YT, MNS, these all are different types of blood groups which are prevalent. So basically a superficial question can be, they might just skip ABO and just mention Diego, Lutheran, Knobs, Duffy, Kell, RH, what exactly are they representing. So in that scenario, the answer would be straightforward, that these represents the different type of blood groups which are prevalent. Even though from a qualitative point of view, you should be thorough with ABO and RH. In PCS, the last time a question appeared that RH blood group is named after which of the following antigen. So basically RH is nothing but the rhesus antigen. Typically, whenever someone asks you what is your blood group, the common replies are A plus, O negative. So basically the positive negative sign which you are attributing to blood groups is associated with the Rh factor. It's representing the rhesus antigen, because for the first time this antigen was discovered in rhesus macaque monkey, the common monkey which you see in the, you could say this, Asia. So coming to the main part, which is the technical part, that how did this blood grouping, especially the ABO, which continues to be the universally used paradigm, came into being. So the credit goes to Landsteiner. Landsteiner was the person who just deciphered this ABO blood grouping at global level. So from the scientist point of view, at times examiner tend to confuse this name with Ian Harvey, who discovered the blood circulation. So make sure you are able to distinguish that blood grouping is ABO by Landsteiner, whereas blood circulation is by the William Harvey. Now the question mark arises: what are the different type of blood groups? How are they named? Why are they named so? So basically in biology, there is a typical concept of antigen and antibody. In immune system, whenever some foreign particle like bacteria, virus, pathogen, toxin enters our body, so that foreign particle is commonly referred as antigen from immune system point of view. Whereas our body's natural immune system gets triggered by the entrance of that antigen, and our body produces some useful proteins which are used to counter that antigen. So basically that particles are referred as antibodies. So basically you need to understand, in case of blood groups, the antibody concept more or less remains the same. Antigen concept is used in, you could say, just for the representative purposes, because when these blood cells, red blood cells, were being observed under microscope, they were tend to be associated with glycoprotein complexes on their outer layer. Since they were present outside the body, so that term antigen was used in that particular context. So you have to understand that when a blood group has A antigen, it would be referred as A. If it has the B antigen, it would be referred as blood group B. So basically, when the straightforward question comes, how are blood groups named? So the key word would be the antigen. So there are different type of antigens as we highlighted in the last slide. There are numerous permutation combination possible, and based on presence of antigen, the blood group are named. So you can say, as you could see on your screen, the first part is signaling the different type of blood groups AB, and O, and the lower column is denoting the antigen part. So in the A blood part, you could see the A antigen is present. In B, there is structural difference and the combination is different. So it was referred as B. And the key point to note is that in case of AB, there is no third antigen like AB. It's just that A and B are present simultaneously. So when both antigens are associated with outer surface of red blood cells, that person is said to have AB blood group. And then intriguingly, the O is not representing the alphabet O. In English, it's representing the nil segment. So basically, if there is a red blood cell which does not carry an antigen, it is referred on the surface layer, it is referred as the O blood group. And this is the scientific reason why O is universal donor, because it does not have any particular antigen. There would be no antibody response from the different blood groups. So that part we will come into the next slide as well. So but just for your information, I'm highlighting that you need to understand that each specific blood group is named after a specific antigen present on their outer surface layer. Then coming to their antibody part, so basically any blood group is, you could say, scientifically programmed to accept blood group of their own type only. Whenever you will try to mix some other blood group, for example, a person is having blood group A and you are trying to, you could say, send a blood group B, there would be clumping because there is an antibody already present which will reject any other blood group apart from itself. So that's where the antibody concept comes into the picture. So basically, in case of A, the antibody B would be present. In case of B, the antibody A would be present. In case of AB, now important part is no specific antibody is present, and that's the scientific reason why AB is considered as universal recipient in the blood group parlance. And O surprisingly has both the antibodies, so that's where the peculiarity will come into the picture. Whenever O has to donate, it is easy to donate because it does not have antigen, but whenever it has to accept particular blood group, only O person has to receive because it does not have the, you could have, it has the both antibodies, so it will directly react with the AB and AB combination. So that's why the person with the O blood group, they at times get a lot of, you could say, complication during blood transfusion. Generally, the donors are not easily available as per the requirements. So I hope I have made clear what exactly is the ABO blood grouping, and when you associate specific antigen RH with them, the nomenclature would become A positive, B positive, O positive or O negative. And you will need to understand this RH antigen is quite common antigen. It is present in almost 85% of population. Once again, consolidating that view that human beings likely evolved from the apes and the monkeys, because this specific antigen is common in the primate order. And you need to understand the persons. So basically, the 85% of the population, even in you are watching, you will see that you are having a positive blood group in the antigen. Now coming to the next level of questions, when they are done with the basic question whether for the blood groups, what are the antigen, antibody, the next level of question revolves around peculiar cases where they will mention 2 or 3 individuals and they will try to frame a question that which person can donate blood to a particular accident victim or a relative, or this person is prohibited from donating in the given set of questions. So basically, such questions are quite elongated in nature. They carry long statements, but when you will read them patiently and use this particular table, you would be in comfortable position to attempt them, because if the question is just revolving around universal donor recipient, more or less 90% of students are likely to crack that question. But if you want to get edge in this particular section, you need to have the right permutation combination available with you. So basically, in the first blood type, the antigen, as we are re-emphasizing, A and B are present. So this person can donate blood to ideally is particular ideal group A and also to universal recipient. So basically, this is more or less reiterating which we have just discussed. But when a person would meet an emergency and he will require to receive blood, then he will once again prefer from this ideal blood group A. Just in case the A blood group is not present, universal donor can be, you could say, relied, because it does not carry any antigen, and the antibody B which it has, it's not going to react with the O. In case of B, the story gets repeated more or less. The B blood group is named after B antigen. It has A antibody. It can safely transfuse to the original blood group B to universal recipient. At the same time, when it will meet emergency, it will rely on its own blood group B and also on universal donor. So basically, if you are clear about A, B is not going to pose much of a problem. In case of ABNO, you need to take a little pause and look at the combination carefully. The first and foremost, as I said, the antigen are not something new like AB. It is just carrying the original two antigen A and B simultaneously. So that's why the A comma B is mentioned. Then coming to the antibodies, once again, it does not contain antibody, which makes it universal recipient. Now, such safe transfusion angle is important one. The AB blood group can safely transfuse only to its ideal blood group AB. The problem why it's only able to transfer to AB, because it's carrying the both antigen. It tries to give blood to B, A blood group person, the B antibody will react. If it tries to give it to B, the B antibody, and O is the worst possible combination, there will be direct clumping, so it will be rejected. In case of the recipient part, all blood groups are present, AB, and O, because it does not carry the antibody. And finally, in O blood group, it does not carry any specific antigen on its blood surface, but it has both the antibodies, which makes it the universal donor. So all blood groups are equally recipient to O generosity. But at times of complication, the O1 will have to rely only on O blood group because it has both antibodies. So basically, during examination, even if you are not having a science background, it's always safer to draw this table and then try to solve that particular question. The antigen antibody part is clear, you have universal donor recipient part, only the minor part needs to be mucked up. So once you decode this, even some lengthy reasoning type question will also be in your grasp, and that would give you an edge of 2 marks, which is quite crucial in state services as well as in UPSC examination. And this template follows in almost different blood groups as well. So you need to just understand the basic criteria of antigen antibody and why it is always preferable to match the blood groups first and then only transfuse. Apart from the other risk of, you could say, there are blood-borne diseases like AIDS, hepatitis, which are once again screened because that is more likely to get other complications as well. Coming to the final part of this section, there is a current affair on blood group. We discussed in our last, you could say, the offline current affair class as well. So basically, if you are attending civil service Gurukul current affair classes, they will definitely give you an edge in examination as well. But in case you have been an old student, you are not able to devote much time, or you visit Chandigarh, it's always better to at least rely on this online, you could say, platforms for at least covering up few important current affairs. So basically, we discussed the crib blood group. It was recently in news. Though even though the crib word is quite pessimistic in nature, generally in English sentences, we tend to use this word when someone is trying to get its unusual demand fulfilled by cribbing over and over it. Typically, some children for a toffee or a chocolate at times crib over period of time. So, but here it's just the acronym. CRIB represents the Cromer India Bengaluru, and the name itself is important because it's related to India. Just like the Bombay blood group is a real blood group, associated with Indian subcontinent. Similarly, this one is quite a rare blood group which is recently discovered during, as you could see, there were 38-year-old woman from Bengaluru, and the UK scientists have collaborated this evidence with rationality. So you just need to mug up that the International Blood Group Reference Laboratory UK has recognized this officially as the new blood group. It is named after antigen, just like the A blood group was named after antigen B. It's representing the CR, you could say chromer antigen present on the blood surface. And this one is the one of the rarest. Chromer system represents the DAF, you could say decay accelerating factor protein on red cells. In simple sense, they're just like we are seeing in red blood cell, there were typical antigen present on their surface. In similar way, there is a specific protein DAF which is present on the surface, which makes it a different blood group as from ABO. So this person has to rely only on the chromer blood group during an emergency. Otherwise, once again, the complications will arise. So in such cases, just as we mentioned in our first slide as well, the blood grouping holds important, and each specific blood group relies upon its own antigen and the specific blood group. Only then there is no clumping. So this basic topic holds importance because at time there could be some accident or there could be some unusual tragedy where there is need of immediate blood transfusion, and the basics of blood transfusion should be clear. Even though this topic is more or less carried out by the health authorities, but still a basic knowledge from administrator point of view, it will present you in a better picture as compared to, you could say, if you are totally ignoring the theme. So in short, we can say, I hope there you have got a basic idea about the CRIB new blood group, which is associated with Bengaluru state location, and then the ABO blood grouping basic template question. Apart from that, you should ideally go through the different type of blood cells, red blood cells, erythrocytes, white blood cell leukocyte, or you could say platelet thrombocytes, and the basic idea what plasma is. In these particular topic also, a lot of factual questions appear again and again in state services. You should focus on the shape of their cells, what is their lifespan, what is their normal count, and more importantly, what are the formation, which is the site where they are destroyed. So these basic themes are given in almost all textbooks as well as your class lectures. So make sure you are clear about their ramification, because we have seen the monsoon season is on, and likely there would be a spurt of once again the dengue cases and the platelet count drops. And typically we have seen during the examination cycle in October, November, such questions then tend to get framed. What are the basic functions of these cells? What are their nicknames? What are their problems? So basically, blood is a comprehensive topic where you should prepare and invest your time considerably. You will likely get the dividend of the theme, and this is one of the conventional themes which is more or less specified in your syllabus. So if you have any specific query which has not been addressed in this particular booster lecture, feel free to ask. I know only few of you are watching, but that's not the whole idea. This theme is mainly catering to some distant students who feel that this online platform is beneficial, and we will bring back another current affair and static topic shortly. So stay connected on the playlist and keep watching. Thank you very much. Have a nice day.